these mature cells here in our body, there are decisions being made. Turn this gene on, or turn this battery of genes on, and turn this battery of genes off. So for example, a muscle cell here, which I think we all would know is very different from a nerve cell, this will have a whole battery of genes turned on that will specify muscle. And not only specify muscle, but what kind of muscle. We have 3 kinds in our body. This one will have those genes, but they'll be kept in an off state. But it'll turn on other genes that say specify nerve and a nerve cell, neuron. And not only that, but what kind of neuron. And then you've got other genes saying, okay, yeah, you're going to be a neuron in the brain, or no, you're going to be a neuron in the big toe, or you're going to be a neuron in the spine. So those also have to tell them where to go as they start to move around here. A lot of anthropomorphism here. They know where to go, how to get there, and they know when they've gotten there. When they know when they've arrived, they stop moving and they sort of set down roots, you might say. But what we're finding is, in this whole area of going from here to here, all this is called developmental life. And it's all really about gene regulation. But what we're finding out is that there's a whole lot more to this regulation than that. This epigenetics thing is starting to come out. And we realize now that, man, there is things that are not encoded in the DNA, but there's information that basically does this. And because if you look for it in the DNA, you're not going to find it. So, from some definitions, first of all, a promoter. Have everybody heard of that term? We all know what a promoter is, somebody that promotes something. Yes, same thing. But here, a promoter, here's a beta-globin gene, is one of the hemoglobin proteins. And here's the gene itself. But up here in this regulatory region, every gene has in its 5' end, or one end of it, a region of nucleotides where they control the rest of it. And this is the promoter. This, our RNA polymerase number 2, transcribes, is this process here, that gene into an RNA. And so this is regulated up in here. This is called the promoter region because it promotes transcription. Okay? So here's where regulation is going to take place. But not necessarily right here. We could be talking as part of the promoter, other sequences that are what we call elements, maybe 1,000,000 bases away, maybe even on a whole other chromosome, that will influence what goes on here in the making of the transcription of that gene. Okay? And all that's epigenetics.
Incredible. Along that same line, I come out of a computer background like Klaus, in aerospace engineering as well. But in my later years, I got involved with computer networking. And to me, that looks like a string of information that's sent that codes for where something is going and where it's coming from and what it's supposed to do and
that's
what
exactly
part of the
what it
computer
is.
it's supposed to deal with. And
That's exactly
incredible.
what it is. Yeah, it is exactly. Now, beta-globin, this is found in part of our hemoglobin. Now, I think we know that of all the cells in your body, which ones have the hemoglobin? Red blood cells. Now, does that mean brain cells don't have this gene?
No, it's not noted for it.
Well, they have that gene.
Yeah.
Every cell in your body has that gene, but only red blood cells actually transcribe the gene. So it's maintained in an off state unless there's something goes wrong in most other tissues. Okay. Here's an example of one promoter of this our metallothionein gene. Fickle. Okay, metallothionein, it's a regulatory region. This is a promoter. Now, promoters aren't just a few bases, but here's where we actually start making the RNA here. From this point, it's called +1. We move on down. So these are DNA. The little E on the end stands for element. That's a term used for a particular sequence. You say it's a DNA element. And a whole bunch of different proteins. These are all protein transcription factors, different kinds, bind to different sequences or elements here in the promoter region. Now what this illustrates is the principle of combinatorial regulation. So just one of those proteins or a couple of those proteins would not be enough. You have to have all of them there in their proper positions to work together before you begin to transcribe the gene. Now just think of it. Each one of these is a protein. That means they have genes somewhere else, which have promoters and have to be regulated. Okay? So now you take the products of all these genes that are scattered throughout the genome, bring them together, and they control the expression of this particular gene. And they all have to be there at the same place, at the same time, in the proper proportions. It's referred to as combinatorial regulation. Okay, chromatin. I've mentioned this before. There's basically two kinds of chromatin. What we call euchromatin, which has an open conformation. Euchromatin is chromatin that's actively being expressed. In other words, you're doing this. Okay. Then there's heterochromatin, which is tightly compacted and closed, and you're not doing that. You're not allowed to do that. Okay. And that all has to do with changing the chromatin structure. I put this picture here because I like it. So that is some heterochromatin. This is what these little colored regions here, we'll look at those more carefully in just a moment. Those are your histones. And you see the DNA is wrapped around the outside of those histones and then woven back and forth. This would be considered a heterochromatin, a closed conformation. If there's any genes in that, they're not going to be actively expressed. Okay? You have to open it up. Now here we have, again, this is sort of a man-made drawing of what you just saw. Compacted DNA with these green balls of histones. You take a look at a histone here, we'll see it again. It's actually an octamer. There's 8 of these things. There's 2 of these, 2 of those, 2 of those, and 2 of those, at the top and bottom here, 2 halves. And that's called the core, or the histone octamer. But what's really interesting, these tails, if you will, that stick out or radiate out from this core here. This whole structure, histones plus the DNA wrapped around approximately twice, is called a nucleosome. And so here's a tightly compacted heterochromatin situation. Here's more open in between. And we have, again, histochromatin, all these other, we've got histones and a bunch of non-histone proteins as well that have regulatory roles. Here again, condensed versus open or uncondensed chromatin. In other words, heterochromatin versus euchromatin. Now, here's this sort of represents that purple and gold figure I showed back a while back. And you see the histone cores here, the nucleosomes, and their little tails. These little red balls represent methyl groups, little CH3 stuck on them. Whenever you have that, you tend to get this structure, closed up, tightly compacted, heterochromatin. When you remove those and add to it a different kind of group called an acyl group with these little green balls, this structure opens up like this. And genes that are now in here are now accessible for transcription factors to do this. Incredible. Now, we're going to see going from here to here and back. And notice the double-headed arrow. We can go back this way. So there are certain times in your development you'll have it this way, and other times you'll have it that way. And whichever way it ends up being when you're an adult has to be maintained that way, too, for the rest of your life. If it's not, then you get problems. Huh? Sorry?
All we have to do now is figure out how we can hold it.
Well, sometimes you want to do that, sometimes you don't want to do that. That's the thing. And here's an actual electron micrograph of, we call it the beads on a string thing. Each of these little white dots is a core histone. Eight proteins there. And the stuff connecting right here, that's the DNA. This is a very open conformation here. And we just call the beads on the string conformation. So that's what it actually looks like. Now, here again, here's your heterochromatin, fairly tightly. It is always put in these what they call radial loops as well. And then these are twisted, not shown in the picture. These are highly contorted. But if there's any genes in here that you want to express, then you've got to somehow go from here to here. You've got to open it up and generate this sort of beads on a string type of comfort, which you just saw. Okay? And then when you've expressed the genes that are in here, then you close it back up, go back up here. Okay? Histones and Nucleus, we've already seen this a little bit, but here is again a computer-generated model based on X-ray crystallographic data and stuff like that. The colored here are the histone proteins, the H3s and H4s, H1s and 2s, or H2s, A and B are down here. And here's the DNA. You notice it's wrapped around on the outside. But here, again, these tails that are radiating out, that's where we're mainly concerned with now, was modifications of those tails. Here's just another picture of two different views of the nucleus, one like we just saw on the side view of it, like that. And again, you can clearly see the tails radiating out. Now, these tails, as I alluded to earlier, are highly amenable to modifications. Now, what was shown here, a little red ball, is a methyl group, which is a little CH3 there. And we can see that different proteins, these numbers here represent positions starting here and counting this way. So amino acid number 1 will be there. So number 4, number 9, number 27, 38, 79, etc. These modifications that take place on these histone tails take place on certain amino acids there. Not all of them, but certain ones. We'll see in a second. And each one is amenable to a binding of a different kind of protein. They can recognize, like CHD1 and BPTF, recognize a methyl group on position 4, but not these, and that leads to transcriptional activation. But these guys, HP1, we'll see in a minute later, recognize methyl groups on number 9. And that means silent heterochromatin. Those convert back to heterochromatin. So there's a code here within the genetic code. And people are still trying to work it out. Okay, DNA methylation. Now here we're talking about the DNA. I just mentioned the histone methylation, and we'll come back to that. Right now it's DNA methylation. Okay. That can be brought about by two ways. Either a protein comes and binds to a particular element and methylates. Okay, fine. Or an RNA, what they call a non-coding RNA. In other words, it doesn't code for a protein. Have you ever heard of the ENCODE project? The ENCODE project? Now that we've sequenced pretty much the entire human genome and many other genes as well, they're going back through that entire sequence and finding out, okay, where are the genes, all this kind of, and they found, much to their surprise, well, I should say much to their chagrin, that virtually all of our DNA is transcribed into an RNA of some sort. And very small portions actually translated further into a protein product. But so the big question, what's all this RNA? That's energetically costly to do that. But yet it's done. And now we're finding out that, golly, all these different RNAs, some of them are embedded in other genes, nested in other genes. They have a regulatory control. They're regulating this entire genome. What goes on, what goes off, and where and there. So RNAs, we'll mention this in a second, again, can lead, now here it looks like the RNA itself is bringing this change about, but no. It actually comes here and then it recruits, and that's another term we throw around rather loosely, it recruits this or that. Now as people, we know what we mean by we recruit somebody, we go out and get something. But how does a protein or an RNA in a cell recruit something? You know, that's, and how does it say, come here, you, come here? We don't know. That's a whole other field.
What on the left side, what's the transcription factor? Yeah.
Transcription factor, these are proteins.
Oh, proteins, okay.
That, you remember that one color, kind of too far back.
That answered my question.
Okay, they're proteins that, so what I'm saying here, a protein can lead to this modification or an RNA can lead to the modification. And here is the modification on DNA. Wherever you, not wherever, but oftentimes we find these methyl groups attached to a C. This is a cytosine. And it's attached at a particular position. These numbers here give you the orientation of the two strands of DNA. They're 5 to 3, 5 to 3. They're anti-parallel. And we have these guys are in clusters all over the genome, and they're called CG islands, or what they call actually C. Well, it's not shown in the picture. They're called CPG islands. Okay, the P here represents the phosphodiester bond that's holding these two together. Okay, and for simplicity, we don't show it there. But that's called a CPG, that's part of a CPG island. Now, not all CPG islands are modified this way. 70 to 80% are, but the other 20 to 30% are not. And they seem to be protected from this modification. In other words, you don't modify. We find these types of structures of this type that is called a dinucleotide in the promoter regions or near the promoter regions of genes. And so take home group here, I took a take home message here in a second, but here's the actual photograph picture of it. Here's a normal cytosine. At position number 5 here, we put this. And so here's our 5 methyl. Now, here's a 5-methyl cytosine. Now, it can undergo spontaneous deamination where this thing comes off. And when it does, it's converted to a whole different base, a thymine. Okay? Now, that's called a transition. That's another kind of mutation. So you can get the purposeful modification, but because we live in a fallen world, that part of that modification can be changed, and now you've got a whole other nucleotide. And you can imagine that that can begin to change the DNA sequence a little bit with these mutations. Where was a C, now you've got a T. Okay, and that can have repercussions down the line, which we won't talk about, leading to cancer, possibly.
Yeah, well, one of our granddaughters has something called Patellas or Achillean syndrome, and it was chromosome 12, 12 or 21, where there's a mutation on that chromosome. And it expresses itself in certain features. It's actually worse than Down syndrome.
Yeah, it's it just shows you how fearfully and wonderfully
Yeah.
we're made, you know, because it's life full of things. Sickle cell, for example, just one letter change, and you got that problem. Now this, once you methylate this on the 2 strands, which I had a minute ago, there. Now when these 2 strands are separated and they to generate new DNA molecules during the process of what's called DNA replication, this strand will have that. This strand will have that. The new strand for this will not have that. The new strand for that will not have that. So we'll have the 2 double strands, but only one of those 2 daughter strands will be methylated. So, but you have to maintain the methylation pattern from one generation to the next. So we have another. We'll show here. Here we have here. So now here's the new daughter strand, double helix. Here's another new daughter double helix from here. This strand is not modified, but we have another enzyme comes along, it's called maintenance methylase, that reads this and says, okay, there should be a methyl group here, so it adds it. And so we're back to where we began, over here. Okay. So that's how once these modifications are done, they can be maintained from one cell generation to another. And if they're done, and we'll see some other modifications, if they're done in the germ cells, sperm or egg, then they can be maintained for the next several generations.
Is this error checking, sort of? Is that what this is
No,
happening here?
no, not
It
this, not-
senses that it doesn't have an ME, so it adds one?
Well, okay, I guess you could look at it that way. Yeah, it looks like, okay, this is what they call a hemimethylated state. And so it says, okay, this is a methyl group here, therefore this should have a methyl group, and it adds it. So if it doesn't, then you can have a problem. If we don't go from here to here by the next round of replication, then you've got a problem. Then it becomes a mutation. So, one of the take-home messages, in general, methylated or hypermethylated CPG equals gene repression, turning on. Unmethylated or hypomethylated CPG is active transcription. Okay? And again, we're talking about the DNA. So that's just one of those little take-home things. Okay, 2 ways methylation can influence transcription, this process here. Here's a little CPG island. Here's a promoter here. This is nearby. Now, this could be hundreds of thousands of bases in here. And the picture looks like it's just right next door, but there could be hundreds of thousands of bases away. But normally here's a protein can bind to this when it's not methylated and bring about some activity, let's say transcription. But if something comes along and methylates it, then this can no longer bind. So this protein, this activator protein, cannot do its job. So whatever gene is here is now off. That's one way. Alternatively, you've got a CPG island, it's already been methylated. It attracts other proteins that recognize this methylation pattern. They bind to it, and then they recruit other proteins to this site. And these other proteins, well, in this particular case, is a histone deacetylase. So here, a modification of the DNA leads to modifications of the proteins, the histone proteins that this DNA is involved in. So the two processes are linked together: modification of the DNA itself and modification of proteins that interact with that DNA. Okay? Here's, for example, here's a specific example of this particular EGR1 transcription factor. Normally, here's like a little CPG island, and it's not methylated. Normally, when this binds, you're going to get whatever is going to influence a whole bunch of different genes. Those genes will be turned on. But if you just methylate this, then it can't bind, and then those genes will remain active or inactive. Here's another example here. Here we have a methylated DNA. It recruits this particular protein, MECP2, don't worry about that. That recruits a histone deacetylase, which removes off the histone tail, removes acyl groups, and then it recruits histone methyltransferase, which adds methyl groups. So it takes off acyl groups, adds methyl groups. So now this reinforces this, and this reinforces that. So we go from a gene that's turned on to a gene that's turned off. But it involves a whole bunch of different proteins.
Wow.
And you see how modification of the DNA and modification of the protein itself are linked together through these various proteins. Again, another take home. Thus, methylated DNA tend to recruit protein complexes that will reinforce the silent state. Remember, methylated means silent, it means turned off, and they're going to recruit proteins that will reinforce that. DNA and histone modifications are linked, as we just saw. So hypermethylation of tumor suppressor genes. These are genes, we actually have genes in our bodies that their protein product tends to suppress the growth and formation of tumors. Okay? So, but if we happen to have a hypermethylation state, remember methylation means turn them off. Hypermethylation of these particular genes, less tumor suppressor, leading to cancer. Alternatively, not enough methylation of an oncogene. We also have genes in our body that when something goes awry, they tend to lead to some form of cancer. These are genes whose protein products function normally in regulating the cell cycle. You know, division of the cell into 2 cells, into 4 cells, and on down the line. But if you, and again, they're controlled too through methylation. If they're undermethylated, then you can have overexpression of a normal good gene. That too can lead to cancer. Okay? And so, yes, there's direct links here to cancer, this type of stuff. Now, another problem, has anybody ever heard of Fragile X syndrome? It's a mental retardation state. Here's a picture, here's an X chromosome here, and always at the bottom there's this constricted, a very noticeable constricted site. And that can continually break off because it's so constricted, it's very fragile. And when that little piece then gets lost, and any gene that's on there gets lost. Okay? And now, this little region right there, here's what it normally looks like. We've got normally 60 to 50 repeats of the CCG. CCG, 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 50 times or so. But over time, those repeats can be propagated, if you will. So we have anywhere 50 to 200 repeats. Okay? Because it's a highly repetitive sequence, highly repetitive sequences tend to be made more of them, so to speak. Made more of them. Well, here, methylation of this, no. Methylation of that, no. But when you get above 200 repeats, you develop this syndrome. You get methylation there because it's a CCG, and the C's get methylated. And any gene, this particular gene is a Fragile X mental retardation protein, is not expressed. because this region has now become hypermethylated, where it wasn't hypermethylated in these two situations. So when you don't produce that protein, you get mental retardation. So all of us, we all have X chromosomes in our body. And so we're probably right in here, but over time, the longer you live, the tendency can go from here to here.
So is this condition something that always shows up at birth, or is it later onset?
It has what they call the time of onset. It shows up later in life.
Mhm.
You start out with a baby, you notice probably pretty much normally. But over time, with every round of replication, these tend to increase in the number of repeats. And once you get above a certain threshold, then that's when problems start to happen.
Could this
This is
be,
true for a lot of different genes, not just this one.
could this be like dementia and Alzheimer's?
Well, we'll talk about that in a minute. Yeah.
Sorry?
Yeah. But it's all related, it seems like. Yeah.
But this could be a reason why older adults tend to have a higher percentage of mental retardation in their offspring. True or false?
Older if they are reproducing when they're older.
Correct.
Okay, is that what you meant?
Yes.
Yeah, okay, let's say a 50-year-old man versus a 30-year-old man.
Right.
The 50-year-old man is more likely to have already more repeats, and so he's donating those more repeats to the next generation.
Right.
And the next generation is just going to add more repeats to it. So, yeah. Okay, now here, closed chromatin equals gene repression. Open chromatin, gene transcription. Okay. Now, here there's an example here of two things here. Now, you can have genetically identical individuals that look phenotypically very different. And what I see here, if I recognize this one with a 68 on it, or maybe 89 when I read it, this is a queen bee, queen honeybee. And all these are other workers. They're all females, and they're all genetically identical. Yeah, clearly. This lives about 5 years, produces about 2,000 eggs a day. These live very short lives, don't produce any eggs. These are sterile. Now, originally, when these guys were all larvae, they were fed this substance called royal jelly. Have you ever heard of that? Okay, royal jelly. They were all fed that. But at some point in time, only one of those larvae was continued to be fed royal jelly. Those would develop into queens and all the things that are necessary for a queen bee. Those that had the royal jelly stopped developed into worker bees. They're all female, they're all genetically identical, but clearly phenotypically very different. Now there was something in the royal jelly that said queenness. And what turned out is there's a chemical in the royal jelly that inhibits an enzyme that adds methyl groups onto the DNA. So the DNA, so there's a lot of genes in here that are on because they're hypomethylated. Those same genes are off because they're hypermethylated. Because the royal jelly had the inhibitor of that methyl. She got the inhibitor, they did not. And look what happens. A whole battery of genes are left on to develop the queen. Now, how many people know, jump ahead again here, Jack, but that's okay. Twins. Monozygotic twins are identical twins. Okay, they came from one egg that's broken. They're broken in half for some reason, and now you've got two individuals growing, but they're genetically identical. Now, early on in life, you see the two twins, and they look like, oh, you can hardly tell them apart. But as they get older, have you ever noticed that you begin to be able to tell them apart? That's because of the epigenetic changes that happen during the course of their life. Okay? So even though their DNA sequence is identical, but yet they don't look identical later on in life, it's because they're suffering from different types of epigenetic changes.
You know, that's actually a very easy way to explain this to someone that doesn't have a lot of scientific background.
Yeah. Yeah, the
That
DNA,
explains it perfectly. There's a
DNA and
picture that
histones
they
are
have.
being modified in a certain way in this individual, but not in that individual. And it leads to these minor differences. Okay, histones, histone variants and their modification. Again, we've seen this picture before. Here's some of the modifications on one of the tails, H3. H3 seems to be modified a lot. H4 can be modified, H2A can be modified. All of them are amenable to some sort of modification. But H3 and 4 seem to be modified a lot. Here are a bunch of modifying complexes that's far been identified. And for example, here's an acetyltransfer. All these complexes, now these complexes are groups of proteins, not just one, but groups of proteins. They will add acetyl groups to a histone tail. Well, here's a bunch of different complexes that will take those acetyl groups off. Here's other complexes that will add methyl groups, and here's other complexes that will take those methyl groups off. So you have complex batteries of enzymes that will modify the histones this way or that way. So on view. But they don't modify in the same place, too, necessarily. Just so here. Here's just a representative example. Some of the modifications that have been noticed in these different histones here. We see this, these are the one-letter abbreviation for each amino acid. This K here represents lysine. Now, lysine seems to be modified a lot. So this lysine at position 4 on H3, and this one can at position 9 or 27 can be either acetylated or methylated, but not at the same time. The little yellow circle represents, now these are usually S's and T's or serines or threonines, can be modified with a phosphate group. So you have methyl groups, you have acetyl groups, you have phosphate groups. But again, the key here is not so much as an individual modification, but the combinatorial modifications. You have to have a modification in this position, a particular modification, and you have to have another particular modification in that position, in this position. That then is translated by the cell to mean this, or that. So it's a particular combination of modifications that matter.
Because it shapes, and the shape determines what protein it attracts.
Yeah, in a lot of ways. Let's say a lot of proteins are attracted to these modifications. And so here we have just a nucleosome with histones here, and acetyltransferase adding these little acetyl groups. And notice that this picture, it's not that much, but it's looser, it's open up here a little bit more. And so when we go this way, we make a more open chromatin structure, more amenable to transcription. And when we go this way, we more closed, less amenable to transcription.
Now, am I seeing accurately the handedness of the spiral change too?
Well, you caught that, okay. This, by this moving around here, this opening, some of that happens. These things here these guys here can, what they call slide, and cause the looping of the DNA to change a little bit, and maybe twist a little bit, so that a gene that's here will now be placed out here. More open and more amenable. Yeah. We'll talk about that in just a second. Here is some acetyl groups, again, these 4 histones here. Some strategically placed acetyl groups, virtually all lysines. There's some arginines, there may be occasionally arginine, that's the A and it's the R, could be modified as well. But here it just shows acetyl groups. Now, again, another take-home sort of message here. Generally, acetylation is associated with an opening of the chromatin and therefore active transcription. Methylation now can also lead to opening, but it depends on the degree of methylation and the location of the methylation of the histone. For example, this is a shorthand. This means H3, that's histone H3, K, lysine number 4, 3 methyl groups. A trimethyl group on that particular position. And this position number 36, lysine number 36, in the same histone, are associated with increased transcription. But methylation of that lysine number 9 on the same histone, and that number 20 on a different histone, are associated with repression. So again, it's not a single modification that makes the difference, it's the combination of modifications. And not just on one histone, but all the different histones at the same time. these are called what's called epigenetic marks. There appears to be a histone code. That is, the combinatorial modification of each histone in the nucleosome confers information which can be translated by the cell. Now, not this translate, but translated. In other words, this modification, or this combination of modifications means to do something. A single modification does not determine transcription activity, but rather the particular combination of multiple epigenetic marks. And here's just a short version for you. Here's the different histones here, 3 and 4, different place, site of modification, the type of modification, and thus far what's translated to be. For example, acetylation at K9 on H3, transcription activation. On H3S10, so serine number 10, phosphorylation, also transcription activation. Acetylation here on that one means prevents. Now look at acetylation here prevents methylation here.
Yeah.
So we're going to see, I'll see that in a second.
So in this twisty wormy thing, you're talking about the 9 thing is momentarily near the 14 thing?
No, the 9 and 14 are, well, they're fairly close proximity.
Yeah. But if this thing is bent and folded,
Yeah, but a
then,
particular modification here will prevent modification over here. But if this is modified, that will prevent modification here. So there's crosstalk when you take a message.
Yeah, and would that be in 3D space? Does that mean you have this chord where the two things that might seem to be far apart are next to each
Oh,
other?
yeah, well, yeah, when you fold and twist and all that, you bring them in close proximity, yeah.
Uh-huh.
Yeah.
Okay.
Just
So
here,
that tells you that the proximity is not really a matter of where things are along a straight line street.
exactly.
All right,
Yeah,
okay.
Yeah. Whenever you see a picture in a book that looks like this nice linear or straight line, in reality, it's not like that at all. Okay. Here again, here's the histone. One modification on this histone may influence a further modification on the same histone. One modification on a histone may influence further modification on yet another histone, as we just saw a moment ago. One modification, here's a histone, and it has a acyl group here. Add another compound, this is like a little miniature protein, attach it here, and that attracts or recruits HDAC, that's a histone deacetylase, which comes to bind to this and then removes the acyl group here, and then changes the activity of the gene. So, it's just, it is. A histone modification here can influence its interaction with other histones, and its interaction with the rest of the DNA. So here we have a histone tail, we have a positive charge, lysines have a positive charge. You acetylate that, you get rid of the positive charge. Here, this will be attracted normally. Opposites attract, learned that in preschool, and still applies. But now when you get rid of that positive, now you've disrupted this interaction here between this nucleosome and this nucleosome. And that's going to have ramifications. But these, again, addition of these groups is very strategic, not just throw them in there, but very strategic. And modification may serve as binding sites for other proteins, regulatory proteins. Now, we've got those standard histones and all their modifications. On top of that, we have variants of the histones. Each histone has one or more variants or varieties. Here, for example, H2, there's a macro H2A, H2A.Z, H2A BBD, H2AX, H2B, that's the standard one. And then we have this other H2B, then we have an H3, a SEN H3, an H3.3, and H. So we have the standards in here, but also a whole host of variants. Now the variants differ from one another, shown here just a linear schematic. The different ones here. You can see just by the length here alone that they're not all the same size. They vary in their amino acid composition. So they're not the same size proteins. For instance, this hardly doesn't have this port, but this one does. It has a truncated one. These two are about the same. Likewise over here, they mostly differ in this, what they call the C terminal. H3 syn, what's called synager, is found at the centromere. This part here, this is a chromosome, and you've all probably seen these figures of this sort of X structure. In the center there is this particular histone is found there. But here, macro H2A is found here in transcriptionally silent regions, like the inactive X, and we'll talk about in a minute. This is also found in regions where we have double-stranded breaks through some sort of haphazard, you break the chromosome. Those then will come in and open up things and help things. H2AZ, that boundary between heterochromatin and euchromatin. In other words, you've got highly compact chromatin, you've got this open chromatin, and if something's not there to say stop, then that heterochromatin will creep into the euchromatin and turn off all the genes there. So you have to have this. And this is also found in double-stranded breaks as well. 3.3 is found throughout stuff. This particular one's excluded from the inactive X, so it's on the active X. So the different variants can be brought into different places, take one of the standard ones out, and substitute in one of these variants, plus all the modifications that go on. Chromatin remodeling. Here's a model of, this is DNA in here, nucleosome. This is the big protein complex out here, in the process of remodeling, or whatever it is. Here are some complexes, basically 4 of them. These are actually together as one complex. So we got 1, 2, 3, 4 different complexes so far. This one has generally been associated with opening up the chromatin for expression. These two have been associated with closing up the chromatin for silencing, so to speak. And this one can be either way. So, normally we have here the histone, the DNA wrapped around it, and here on this order of second, wrapped nucleosome exists for about 250 milliseconds. But occasionally can unwrap and last for about 10 to 15 milliseconds, then it can rewrap. And this is always going on. So these aren't static structures. Okay?
How do they measure this? With an electron microscope, and they watch this stuff going on?
No, you can't, not with electron microscope. You can just,
Or just chemical reaction?
look at, well, you can label the DNA, you can label the histones with fluorescent molecules and do time-lapse things. You can see that and watch it change, go from non-fluorescent to suddenly fluorescent and non-fluorescent like that. And then you take that data and equate it with, you capture it in like an electron microscope. Okay, when it's flashing, when you see it, it's in this open cup. When it's not, but most things is closed.
That technology just is mind-boggling.
There's so much technology out there. It is the technology alone is mind. Yeah. Chromatin remodeling, they can cause, as I mentioned earlier, twisting or rotating of the DNA around the histone, thus exposing different parts of the DNA there that were not maybe exposed here, but now they're exposed here. This can cause a sliding of the histone, so now we displace the histone, opening up this region. Or we can actually just evict the nucleosome, evict the histone, just throw it out altogether, and leave this wide open here. Okay? Or, and the next one here, we can come in here, we've got a histone, but now we can bring in and switch out the standard dimer, H2AB, and put in one of the variants. Or bring in a whole different nucleosome, complete with full of the variants and their modifications. So these are remodeling complexes.
All these variants that you're speaking of here are so far within design processes going as they're supposed to?
Yeah.
Okay. So all of this is happening as designed, and all that has to happen for things like us to stay alive and grow and all that kind of stuff.
For us to develop from a single egg into all the different.
And you know, this and it tells you that there is a lot of room potentially, unless there's some kind of correction that we didn't talk about yet. There is a tremendous amount of room for error to creep in, unless there's something stopping it. And maybe that error prevention is another layer we can
Well, that's another layer we haven't looked at yet.
Okay.
And,
I mean, does it seem, with that kind of complexity and the things happening with exact timing and so forth, with complex processes that we know of, such as the working of the computer circuit, you have lots of room for stuff to creep in, because you know, you can't talk about these as simple things. So, you know, because the timing's got to be right, and everything's got to be positioned just so, and you know, the amount of stuff that has to be right in order for it to work at all, you know, is irreducible complexity.
yeah, exactly. And it just also just further illustrates the point made in the Psalms, how fearfully and wonderfully we were made. You know, when you start to really analyze a single cell on this level, and then there's going to be deeper levels that we haven't gotten to yet. This is going to open up other questions, which is going to cause people to do more. Okay, well, what about this? You know, how do these remodeling complexes know to take this out and substitute this particular variant in? And when? And where?
Now, all this stuff has to be going right at a frenetic and complex pace in order for us to, on the physical plane, keep our thinking capacity. Okay. Which goes to say that the faculty we have to hold this conversation is, with what we've seen so far, extremely delicate.
Yes.
Okay.
We are precariously perched on the edge of eternity.
Yeah, okay, you know, and if we imagine that our faculties are extremely robust and durable, then we might be looking at the Nebuchadnezzar syndrome, which has probably not been documented yet.
Well, I was thinking about, our pastor has a Down's baby, and she's about 4 years old, but she's very small. I mean, she can walk. She's finally caught on to walking, hasn't she?
Mm-hmm.
And she's adorable, but you were talking about growth rates can be affected by these, and it must be that somewhere in the Downs.
Well, Down syndrome is due to, there's a certain little group of about, I think it's about 5 genes on chromosome 21, not the entire chromosome. So there's that little group of genes. If you have too many of them, you normally have, everybody normally has two copies of that group. But if you have a third copy, you get the Down syndrome. Okay? And that can be caused by, there's 2 different mechanisms to bring that about, which we won't go into, but the people that suffer from that. So again, it shows you, again, this is I think, and nobody really, I've not heard anybody mention it, that when you, part of evolutionary theory is that to go from a simple, I'll say simple right now, let's say a bacterial-like cell, single cell, with one chromosome, maybe 1,000 genes, to something like us, where we've got many chromosomes and many more thousands of genes, okay? Something much more complex. Somewhere you have to figure out, you have to explain how you go from one chromosome, let's say, to 46 chromosomes. Where does all that extra information come from? Where does all that extra DNA come from with all the information that's in it? And one of the explanations is, well, gene duplication. Have you ever heard of that? That there was this primordial gene and it somehow duplicated. Now you got 2 copies, but you only need 1 to live. So then you can, since you've got 2, you can, evolution over time can play with the 1, tinker, make a mutation here, make a mutation there, and see what falls out.
The mutations are always bad, aren't they?
Well,
Well,
yeah.
this thing's not looking very fault tolerant. What you said so far would say that if you got too much experimenting with that extra gene material, the critter's going to die.
Well, that's right. That's right. So it has 2 genes to play with.
Yeah.
If it plays too much, the critter dies. But so there's going to be a limit to how much tinkering it can do. But supposedly it'll do enough tinkering, or will have done enough tinkering, to take one of those 2 copies now and generate a new protein with a new function. Okay, then over time, that gets duplicated, and so on and so on down the line. You end up with all these protein families.
It's a
Okay,
fantastic story.
it's a fantastic story, but still, at it, for a long period of time, you've got 2 copies or more when you normally have 1 copy. And from Down syndrome and other syndromes like it, we realize more is not better. You know, it can be very detrimental. Okay,
So what that
that doesn't enter the mind. Yeah.
that Pilaster-Killian syndrome I mentioned to you earlier is also on chromosome
Also
21.
21.
Yeah, and our granddaughter's 4 years old and she can't even walk. She just can barely roll over right now.
Okay.
Yeah.
Yeah, right now, as part of the ENCODE project, is taking all the different chromosomes and mapping out all the different known diseases. Where are they on each chromosome? Which chromosome has the gene or genes that are responsible for this syndrome or that syndrome, and where on the chromosome are they? So once we get that, and then we know the sequence of the good gene versus the aberrant gene, then the idea is how can we go in and fix it? You know, where possible. Now this kind of stuff, we don't know enough about it to go in and tinker with this. No, we want to add methyl groups here, take methyl groups. This has been dealt with some people that suffer from certain kinds of anemia, where you've got, I know I'm getting way off track here though. We've got here some 2 chromosomes. One contains the beta cluster and then the alpha cluster. A product from here and a product from here give rise to hemoglobin, okay? We got 2 of these and 2 of those. But on here are different genes, different betas, different alphas that are expressed in time. In other words, this one will be expressed and this will be expressed when you're in the womb. Later on in development, these are turned off and these are turned off to make a slightly different version of hemoglobin. Okay? And then these are turned off and these are turned off, then these are turned on, and that may be last the rest of your life after you're born. Okay? Now, the ones that are turned off is because, we'll see here, the chromatin has been remodeled due to methylation of the DNA in the promoter regions of these. And so if you've got a problem with these normal adult genes, they're not producing enough hemoglobin, is the possibility, can you go back and turn these on? And would that be a good substitution for a lack of normal adult hemoglobin? Okay. That's a great idea, but nobody's been able to do it successfully and stuff like that. Of course, it's kind of difficult to do that.
Yes, what you're saying in this slide though, are the substitutions that we're talking about here, the kinds of things that are the difference between someone who's tall and short, or someone with black hair versus blonde hair?
No, not necessarily,
small
not
changes.
necessarily. Well, these all will deal with fairly small changes, but I mean, you saw the queen bee versus the worker bee. There's a noticeable change there. So most of these changes would be what you call like maybe minor changes, unless it produces a cancer or something like that. Yeah, but again, yeah, I'll just say that.
Okay.
No small changes. Not necessarily the dark hair versus light hair, stuff like that.
Okay.
Going to here, what I was just saying, for example, here's a chicken. Here's a chicken DNA, here's a globin, and their DNA is organized a little bit differently than ours. We have the globin gene here, adult, embryonic and adult globin genes here. And early on here, erythroblasts here, these arrows here, we have a bunch of these arrows. Those are what are called DNase-1 hypersensitive sites. Now that tells you that the chromatin is open, amenable to be cut by DNase-1, this enzyme. If the chromatin were closed up, this enzyme would not have access, couldn't cut, okay? And so here, in the 24 hours of development, we have a hypersensitive site up here in front of the embryonic gene, okay? And 5 days later, they have a hypersensitive site right there in front of the embryonic. We develop some more sensitive sites, but this one right here in front of it, because that's the one that's going to be expressed. In other words, this is opening up. Then 14 days later, that hey, the hypersensitivity has gone away. So that means the chromatin in this region has been closed down. This is now turned off. But look what in front of the adults we develop an open and therefore sensitive site. Okay? So you sort of follow this over time. In the brain, it looks just like that. There's no hypersensitivity because the brain doesn't produce hemoglobin. So all these genes here are just tightly, they're heterochromatic. Okay? And you can tell when something's become open, euchromatic, or closed, heterochromatic. Open off is on and closed is off by the development of these hypersensitive sites. Let's do that. Okay, these next few are just some pictures. Here's some specific known remodeling enzymes that come in and basically remove acetyl groups. There's this deacetylase and stuff. These are found in yeast. And then usually if you've got an acetyl group here and you want to turn the gene off, this is the active. You have to first remove the acetyl group. You'll have to deacetylase. Then you bring in another protein that adds methyl groups. And the methyl groups then will recruit certain proteins to that methyl group. And then these proteins begin to add more methyl groups and more of the proteins come on, like we see here. And we self-aggregate, and now this is heterochromatic, the genes are off. This is what happens in us. A lot of our genes are controlled by this HB1. In fact, HB stands for heterochromatic protein 1. And it's just another thing here. Okay, we'll talk about this in a minute too here, just real quickly. Two complexes, PRC1 and 2 polycomb, named after what they found in flies again. Here's a section of DNA. There's these regions here. This first complex piece, PRC2, recognizes this, comes and binds, starts adding methyl groups to certain histones here. Trimethyl groups here. That then recruits PRC1, and then you get this knotted formation here, a heterochromatin forming, and you close off the genes. Now, I want to go on here, just real quickly here. We have these things called insulators. And you can modify this in here, keep it open, but on either side, you don't have to have the same modification. You keep them closed. Okay? Now, insulator here is an enhancer. That's what it says. Enhances the product, or enhances the transcription of a gene. But these insulators keep this enhancer from activating that gene, so it's forced to only activate that gene. We'll see that in a second. Now I want to go on, skip over here real quickly. Now, every one of us, for all of our genes, we have two copies, or what they call two alleles. We got one from mom and one from pop. Okay? Now, in there are many genes that what happens is what we call monoallelic expression. You only express the one you got from your dad, or you express the one from your mother, but not both. When you do both, you're dead. So how do you evolve that? You know, but that seems to be pretty random for most of those alleles. Not many of those alleles, but enough of them found in different tissues, very important genes. You express one or the other. But some alleles, you express just the father or just the mother. In other words, it seems to be dependent on where you got that chromosome from. Okay, if you got this allele from your dad and this same allele from your mom, it depends if it's in other words, it's sex dependent. So we always inactivate the one from dad, or we always inactivate the one from the mom. So somehow, the cell has to be able to know which chromosome came from mom, which one came from dad, so we can inactivate the right one. Okay? And not keep them both on. And it does this through a process known as genomic imprint. Now we know about ducks and stuff, how baby ducks, they'll imprint onto you. So what happens here is basically here. You have here this fertilized egg. The two alleles, alleles 1 and 2, and we say differentially methylated. And this allele 1 is methylated, and this allele 2. And methylation means turned off. So this one here's on, that number 2 is on. Okay? So the cell has an on 1 and an on 2, so it's good. Well, it develops into an embryo, and all the other cells in the body will maintain that same methylation pattern. But there are certain cells that become germ cells, sperm array. Okay? So here's in the father, in the sperm, and in the mother. Now, when the father makes new sperm or another new egg, the sex determines which allele is going to get modified. So the mother will modify allele number 1, but not 2. The father will modify allele number 2, but not 1. Okay? And now these get together, and back to restart again. Okay? In other words, the father allele always will do number 2 because that's sort of the male. The mother will always do number 1 here. Okay. Yeah. This leads us to X inactivation. Now we all have X's. Now all the women here have 2 X's. 2 X's. Now there's this thing called dosage compensation, where you only need 1 X, and you're only supposed to have 1 X active. So for guys, no big deal. We only have 1 X. But for women, it is a big deal. Because if they don't inactivate one of those X's, they're dead. Now, the choice as to which one they got an X from their dad and an X from their mom. So which one should they inactivate? Well, it appears to be random. Okay? But whichever one they inactivate, they then have to maintain that inactivation state for the rest of their life. Every cell has to have an inactive X. Okay? So here we have it, in placentas, we'll say this is like us. We have 2 X's. We'll inactivate at about this stage. We inactivate 1. Then you erase the marks, the epigenetic marks that cause this to be inactivated, or released in the zygote, in the germ cell. And then when you start developing to all the other cells in the body through development, every cell initially, very early on, will inactivate one of those X's. Either inactivate the X from the dad or inactivate the X from the mom. But whichever one they inactivate, all the cells that come from those cells will have the same X inactivated. Okay? So you women, every cell in your body is like a mosaic. You have one cell where it's interacted with dad's X chromosome. The cell right next to it may have inactivated mom's chromosome, or X chromosome. Okay? And if you don't do that, and what if you have cells has three X's? Or 4 X's? Nothing happens. Somehow the cell's able to count how many X's it has. You'll know that actually everyone except one of them. Don't even maintain
the right one.
Well, either one.
We'll keep the right one activated.
Yeah, well, the right one here in this case in our case, the right one could be either one, but it only keeps 1.
Okay.
In some animals, it's always the father's that's inactive. Then you have to think about the right one, or it's always the mother. And we'll see an example here.
Well, when you said the word mosaic, that Pilaster-Killian syndrome is a mosaic.
Oh,
They call that a mosaic.
okay.
So it can express itself in different places of the body.
Slightly differently too.
Mhm.
Yeah.
Yeah.
Mhm. So basically, again, what inactivates the X chromosome is all of it, epigenetic effects. So we have two X chromosomes here, and then on there there's this gene. We'll see this in a second. Gene called the Xist gene. Okay, there's several other genes you'll see there. A for active, I for inactive. So initially start out like the same. Then the inactive X starts producing an Xist RNA, which then coats this chromosome from which it came. That will attract this polychrome complex, which will add methyl markers. And this whole thing just clumps up, becomes what we call a polymer. Never to be opened up again. Okay? What's interesting here's that X inactivation region. Here's the gene product that acts on RNAs, on protein, that actually causes the inactivation. Here is a gene on the opposite strand, but going in the opposite orientation. So this will produce another RNA that will be complementary to this one. So on the active X, this is produced. On the inactive X, this is produced. So the active X, when it produces this, you notice this is actually that in reverse. This is what they call an antisense RNA. This will bind to that and keep it from inactivating one of the X's. This gene stimulates production of that by the inactive X. This gene maintains production of that by the active X. So the active X can stay active for the rest of your life, and the inactive X can stay inactive for the rest of your life.
Yeah.
And these are all RNAs. And here's just another picture there. Here's just another showing you things there. Now here, these dark, these are inactive X's. They form what are called bar bodies. Now here is a blastocyst here. Here's the inner cell mass. This is what's going to form the embryo. And this is going to form the placenta, part of the placenta. Blue and pink tells you at this point in time, both X's are active. Okay, the blue is the male, the X from the dad, and the pink is the X from mom. But a little bit later, when in plants, it starts to develop. Notice here, very little blue. The male X, the one from the father, has been inactivated here, selectively, not the mother's. But down here, it's still random. Some are inactive and some are not. And then later on, as this develops into a full-fledged organism, okay?
So this is basically forming the sex, what the sex of the baby's going to be, right?
No, that's already determined at the point of fertilization.
Fertilization. Oh, that other slide that you showed.
That's determined. Now here's one that had 3 X's in it, and it inactivated 2 of them, so it's got 2 bar bodies. And it always keeps one. Yes.
Are they really pink and blue?
No, that's the stain so you can see their activity. Yeah.
That'd be cool if they.
Yeah, these are these are going to be little girls and these are little boys down here.
I was wondering about that.
Here's 2 cats. This is a calico cat. All calico cats are female. And some of the somatic skin from this one here was cloned into this one. This is another little kitten. It's called copycat. So these 2 are genetically identical because the genes from here came from that. But clearly, there's a difference. Again, showing you how genetically identical, but epigenetically different, which leads to differences in appearance. And that's the real name of that little kitten, copycat. Okay. Just real quickly, here's IGF-2R as a receptor, and then IGF-2 insulin-like growth factor. Maternal, mom's chromosome, paternal, pop's chromosome. So these are the same genes on 2 chromosomes, but you can see the mother's has been modified. This ISIS imprint control region is like an insulin. It's been modified, but the father's chromosome has not been modified. What happens is, when you don't have this modified with a methyl group, this gene, this air gene, is produced. Okay? And this air gene will inhibit the production of this gene from this chromosome. If the ISR is modified, then the air gene is not produced, but this gene is produced. Okay? So, mother makes the receptor for this insulin-like growth factor. Father does not. Okay? You've got 2 alleles, but you're only going to express this one. Okay? That's the receptor. 2 other chromosomes have the IGF-2. One from mother, one from father. Now again, this ICR insulator here. When it's modified in the father's, this enhancer promotes transcription for IGF-2. But when it's not modified, this protein can bind there, acts like an insulator. This cannot influence this, but it has to influence that. So mother produces the receptor for this, but father produces that. You have to have the 2 sexes. If you don't, do that, if you don't do it right, there's death. Development just will not happen, further development. Okay? Basically here, in the male, IGF-2 is on, that's a hormone. The female, the H19 is on. And that's on here. Here, another reason why you need two sexes. So here's normal zygote here, you have mother and father, father and mother's nuclei. Get together, get normal development. But what if you had two mothers' nuclei? Or two fathers' nuclei? You got the same number of chromosomes. You got all the genes you need there. But what happens? Development becomes arrested at some point here. And development becomes arrested here. You can't develop beyond a certain point. So again, you have to have a male and female, at least in humans, in mammals, okay, to get proper development. And I think that God designed it that way, through these imprinting these genes. Say, okay, mom's going to produce that gene, but the father's going to produce a different gene, but the two genes have to work together. The products of the two genes have to work together. Can't have one without the other. Other phenomena, just running out of time here, but this will be interesting here. Here is a mother rat, and here are pups and stuff. Maternal licking or grooming. If it's low, if the mother doesn't pay that much attention to her young, the young develop methylation patterns in certain genes in their brain. But if she does give them a lot of attention, they don't develop those methylation patterns. And these methylation patterns in this particular instance has to do with a glucocorticoid receptor, which deals with stress. Okay? And so, baby mice or rats, and probably people as well, who don't get much maternal grooming or attention, will have to develop a certain methylation pattern in the brain, which stays with it for the rest of the life.
I'm going to ask a really sensitive question, Brad, only because I want to take something from this to go home and discuss with my wife, because we've talked about this. But there are some reports that have been written on homosexuality, that it actually changes things in the brain. Is this the process, the
This is most likely what it is, this kind of thing, some epigenetic changes. And you know, there used to be, they look for the epigenetic, look for the gay gene. And they've not found any gay because they're identical. But now this kind of stuff is opening up. Everybody's in the process now of studying what they call the epigenome. We take the entire genome, where are all these epigenetic markers? What do they mean? How do they differ from one individual to another? And there may be, if they look at enough gay people, that they will find epigenetic markers that are prevalent, much more prevalent in them that are not in the straights. And then, of course, they're going to jump on that. So
Well, to me, it's not an issue of whether you're born with it or not, because even if there are biological features and functions that happen in that behavior, it's still a choice. That's what I tell people.
Yeah.
So God's not going to call something a sin that's not a choice. So for some people, alcoholism is a problem. That's a choice, or pornography, or whatever it is.
Yeah.
But it's the same thing. It's still a choice.
Yes.
And if God calls it a sin, it's a choice. Whether you're born with it, or whether these epigenetic things are happening, the bottom line is it's still a choice.
Exactly. All of us here are straight, I would assume, and so we have a propensity, we're attracted to women. Okay? And even though we might be married, we see a young, nice-looking young lady, you want to look at her. Gays are attracted to the same sex. Just as we have to exert control over our natural tendencies, they should exercise control. They don't want to exercise that control.
Right.
It becomes a choice. Right. Sure, they may be born with a genetic propensity for that. That hasn't been shown at all yet, but it may be in the near future. Bottom line,
But it doesn't change things.
It doesn't change things. They still have to exert and recognize it for what it is. Okay,
But couldn't this same thing work between people and animals? We have parakeets at home, and we handle them all the time, and I've noticed since we got the birds, they get tamer by the year, and they're just incredible. The personality they develop and the way they want to be with us, and
I would not be surprised if we can find that our behavior towards animals has an influence on them, especially if it starts out very young,
and theirs
And
probably
vice
has an
versa.
influence on us
and
too.
vice versa, right?
Yeah.
Exactly.
I'm making an obvious point that I think Klaus has already started to make, which is it's risky anyway to tie together physical chemistry with desires,
Yeah.
because there are a whole host of things people are very, very inclined to do that they shouldn't. And, you know, it can be as obvious as me having something popping through my head that's scandalous, and you know, the instinct seems to be like a reflex to blurt it just because I want to have that moment of superiority. And, you know, it is actually murder with the tongue to say something that shouldn't be said. But the machinery is there almost to the point of involuntary. And I believe I was born with that, and the desire is very, very strong. And who knows, maybe someday, maybe some biochemist may identify a brain chemistry associated with this urge to be superior when I don't need to be. Okay, and you know,
You
this
got
is
that
familiar,
problem
right?
too,
Okay. And you know, the urge to commit murder with the tongue is really what you have to
Well,
follow,
yeah,
you
there's
know,
certain things like that.
destroying a reputation on impulse. And so we have to say whether there are physical or biochemical associations with a behavior or not. Sin is still sin, and the
Oh, yeah.
extreme desire to the point of reflex to do it does not justify it.
That's right.
You know, I want to do something powerfully bad at this moment. It overwhelms me. It changes my body temperature and everything. And so, is it now right because I wanted to do it so badly? And
Nope.
And it's gonna be different things for different people.
It's going to do.
Yes.
But it's still a choice. It's
Still comes more down to a choice, exactly. Now, this may be the last thing I mentioned here. Effects of diet. Now, here's a bunch of mice. You can see clearly they differ in their coat colors. But they're all the same. They're all genetically the same. And what happens is certain genes that code for the color of the coat. And on some of those genes, a transposable element, have you ever heard of jumping genes? Jumping genes, okay, not beans, but jumping genes. These are mobile genetic elements that can jump from this chromosome to another chromosome or another place in the same chromosome, different ways of doing that. And they can jump in the middle of a gene and disrupt it, maybe leading to death, or they can just jump around.
you had a ticket number for you to read.
Okay. Read it just a second. Very good.